Chapter 5, Part 1. So just to review, when it comes to cell theory, we've already talked about how all living organisms are going to consist of cells, and now we're going to be talking about how life is going to come from cells. So the ability of cells to generate more cells is through the process known as cell division, and that is then going to support cell theory. So all life, as far as what is considered to be alive, is going to be considered cellular. So anything that has life and meets those characteristics that we talked about in past lectures, that is going to be what determines life. And we know that all cells have to come from pre-existing cells. And that might kind of seem, like duh, but not too long ago, before we had microscopes, scientists believed that life just spontaneously appeared. And the reason that they thought that is that when they observed an item sitting out, so for bugs, for example, they saw them just grow and then come from seemingly nowhere or out of nowhere. And so, obviously, what we know now with microscopes is that in order to have life, it has to come from pre existing life. So, just make sure that you know that there are also some living organisms that are alive and exist just as one single cell. And then there are some organisms that are made up of trillions of cells. So as you can see, there's a single cell on the slide here that would be considered living. And then you can also see with the elephant, even you as a human being, we would be made up of trillions of cells. Cell division is the formation of new cells, and cell division is at the foundation of growth, repair, and reproduction. So for you to grow to be your adult size, your cells had to go through cell division. For you to repair anything that might be broken or injured, you also have to have cell division. And also, in order to have any offspring, there has to be some reproductive organisms that use cell division to reproduce what's called either sexually or asexually. So sexual reproduction means that it will take two parents, and cell division is going to need basically a female and a male when it comes to this in sexual reproduction. So the two parents are going to have variable DNA, and therefore their offspring is also going to have some genetic variability to them. So gametes, which are our human egg and sperm, are going to be formed in cell division in the gonads. And the gonads for humans are the testes and the ovaries. And the only way for them to be able to form is via cell division. So you can see on the right, on the top, there's a picture of a male sperm, and then at the bottom is a picture of a female egg. Both of these pictures came from cell division in the testes and in the ovaries. And within each of them, there's genetically different DNA, which is why you come out having different genetic DNA than your parents. So just to reiterate here, we have the gametes at the top, which are the sperm and the egg, and then they're going to come together and they fertilize. And when they fertilize, they're going to create what is called a zygote. And a zygote, again, is going to be where the sperm and the egg unite. And then the zygote itself, because it's only one cell, is going to go through the process of cell division and end up with many, many cells in the form of an embryo. Then from there, that will continue cell division until it turns into a human, and then the process repeats itself. So the other type of reproduction is called asexual reproduction. And in asexual reproduction, one parent is going to be producing genetically identical offspring. So this means that any of the offspring that comes out of this individual is going to have the exact same DNA sequence as the parent. So there's no sperm or egg involved when it comes to asexual reproduction. Some examples you can see down at the bottom is through an organism called an amoeba. An amoeba is a single cell protist, and it has the capacity to asexually reproduce. It will just need itself, and it copies all of its internal structures, and then it will split apart, which we call binary fission. At the top right is a picture of a sea star. In the sea star, you can see there can regrow its whole body from a severed arm, as long as it also has a little bit of the central body. So that has the capacity, if it loses limbs, to regenerate it. And then down below, you can see in the strawberry plant that the strawberry plant creates runners. that plant new strawberry plants, establishing that way via asexual reproduction. So this would be a great advantage when you don't have another mate to be able to go through the process of reproduction. So none of these organisms here are going to need um, another individual from its species in order to reproduce. So let's talk about the basics of DNA then, which leads us to reproduction. So all life on Earth is going to use DNA as the genetic material. If you remember, the nucleus of each cell is going to have a DNA sequence in it if it is a eukaryotic cell. In prokaryotic cells, it will still have a DNA sequence. It will just not be housed in the structure of the nucleus. So if we are talking about the sheer size of the DNA in eukaryotic organisms, you'll see that it's going to be a very, very long, huge piece of DNA, but it's condensed into a structure that we call chromosome. And each of the chromosomes contains a specific sequence of DNA that we call genes. And genes are considered to be the unit of inheritance. So genes are just a tiny little length of DNA and a small segment of that chromosome. And the purpose of the genes is to direct the body to what it's supposed to be doing. So basically acting as directions. So all eukaryotic cells have a prominent nucleus where that chromosome is going to be stored. So remember, that's just if it's a eukaryotic organism, it's going to have a nucleus. Prokaryotic organisms do not have the nucleus. So different species will have a different number of chromosomes in the nucleus, and you can see on the left side the comparison of different species. We can also see that the complexity of the organism has nothing to do with how much DNA is found in that individual or chromosomes. So you can see at the bottom there that bug is going to have 8 chromosomes, corn has 20, humans have 46, a pineapple has 50 chromosomes, an elephant has 56, and a dog has 78. So those items indicate to us that the complexity doesn't matter. We as humans are probably one of the most complex organisms, and we have 46 chromosomes, and a pineapple, that is just a pineapple, has 50. So how many chromosomes did you inherit from your mother? So if you remember that each of your body cells contains 46 chromosomes, what happens in the process of reproduction is you inherit exactly half from your mom and half from your dad. So how many chromosomes would you get from your mom? We should have gotten 23 chromosomes from mom and 23 chromosomes from dad, because that makes the magic number of 46, which is what we should find in all of your chromosomes in your body. So inside the nucleus, that chromosomal DNA is going to be organized, and it's going to be very tightly condensed. It's wrapped around proteins, and when it becomes wrapped around proteins, it is called chromatin. So most of the time, chromosomes are unraveled in a form of loose chromatin, which is a picture that you see on the right, when it's going to be somewhat unraveled, but you can still see these little circles representing each one of the proteins that has DNA wrapped around it. So when the cell goes to copy the cell in cell division, it's going to copy its chromosomes and prepare to divide. And when it does this, it condenses them and packs them into a dense pair called sister chromatids. And they're going to be joined at the center via a small button that you can see here called a centromere. And a centromere is what you see holding together those sister chromatids. So at cell division, when the cell is going through the process of dividing, the chromosomes become very tightly packed because it makes it more manageable to move the DNA, as well as more manageable for the DNA to separate. And it allows the DNA to become very easily duplicated because it's so tightly packed and condensed together. Cells have regular cycles of growth and division, in that cells are going to live for a finite amount of time. So cells have kind of their own lifetime, meaning that they're going to have a time or a spot where they begin and then a spot where they die. And that's a natural thing that is supposed to happen. So healthy cells can only start dividing if there's a need for replication. So if there's not a need for replication, then ideally the cells are not going to start dividing. If cells go through an unregulated cell division, meaning that they are just constantly dividing without any sort of control, that's typically what's going to be the foundation of cancer, because cancer means that these cells are not going through a controlled cell division and they're multiplying in large amounts without any sort of control to limit the amount that divide. So now we can talk about the cell cycle. The cell cycle is an ordered sequence of cell phases that's going to encompass the lifetime of a cell. In general, there are two broad phases. There's interphase and the mitotic phase. So oftentimes, interphase will be clumped in with the cell cycle, but now we're going to look at it a little bit differently. Interphase is where the cell spends most of its lifetime. In fact, almost 90% of its lifetime. If we took 24 hours for a cell to divide, it would spend almost 20 hours of it just in interphase. So the cell is still functioning, but what's happening is that it's preparing to divide. So even if it looks like it's resting, there's actually a lot of activity that's happening inside the cell. And if we were to grab any collection of cells, we would see most of the time that the cell will be in interphase because that's where it spends most of its lifetime. The mitotic phase is when the nucleus is going to be active. So that's also a key idea here. When we talk about the prophase, metaphase, anaphase, and telophase, we are specifically talking about what is happening in the nucleus. And remember, that's where the DNA is found. So we will specifically be looking at what DNA is doing. So here's a picture of a graph of the cell cycle. So if we consider the cell as starting down at the very bottom of the picture, it's going to be going through the cell cycle. So when you get to the next phase, we'll see where it will duplicate in interphase, and the insides of it are going to duplicate. And then when it gets to the mitotic phase, that's where it will complete prophase, metaphase, anaphase, telophase. But if just looking at this graph, you can see the sheer amount of time that it spends in interphase. So now we're going to look at some characteristics of each of the phases. The first one we'll look at is interphase, because that's where this cell spends most of its time. So during interphase, the cell is fully functional, but what it's doing there is a lot of activity that you can't see from a microscope. So because it's preparing itself to divide, it's going to double its chromosomes, and then it will have an abundance of DNA, and in that process for cell division. So on the right side, you can see an image of the cells that are in mitosis. These are all plant cells in that top picture. You can see because they kind of look like squares. You can see there's a couple of examples of cells that are circled or have a box around them. So the ones that are in orange are in interphase. And so you can see actually most of them look like they're in interphase because that's where most cells spend most of their time. So mitosis is when you have active cell division, and it's going to be occurring in major stages that you have probably heard before. Mitosis is specifically looking at cell division of the nucleus. There's a helpful acronym to help you remember the interphase, prophase, metaphase, and anaphase and telophase, and that acronym is pronounced IPMAT. We can see down below what the cell looks like when it's in each one of these phases. So we're not going to go into a ton of detail about each one of them, but the important thing is that you're looking at the DNA. That DNA is specifically organized in a formation that allows us to identify what phase it's in. So prophase on the left is when DNA condenses and forms visible DNA. So that's how we know it's in prophase. Then we have metaphase, where it's going to line up and down the middle of the cell. Then we have anaphase, where the DNA is going to be pulled apart, and telophase, where the cell is going to be going through the process of dividing. So telophase oftentimes takes the exact same time as what's called cytokinesis, but cytokinesis happens in the cytoplasm, or it's concerning the cytoplasm, and right now we are just looking at what's happening in the nucleus. Cytokinesis, again, is the final step, but it is talking about what is happening in the cytoplasm and not the nucleus. So this means that cytokinesis and telophase pretty much occur at the same time, because telophase is the last part of mitosis when the nucleus is dividing, whereas cytokinesis is talking about the internal parts of the cell other than the nucleus that are dividing. As a result, the two cells are going to be genetically identical when they are done, meaning that they're going to look exactly like the DNA from the cell previous to it. So again, to reiterate, cytokinesis is when the cytoplasm and all the organelles are going to be dividing. So in animal cells, which would be like you and me, when we see cytokinesis occurring, we see an indentation occur from the edges of the cell. That's what we call the cleavage furrow. And the cleavage furrow is essentially that cell membrane forming, so it starts to create an indentation to allow the finality of the cell to separate from each other. Cytokinesis in plant cells, instead of the cleavage furrow, we see the development of a cell plate. And the cell plate essentially forms in the middle of the cell as opposed to the cleavage furrow. So it starts to build this cell wall, and then it pinches apart, and eventually we get the full separation of the cells. So as to this point, what mitosis does for organisms, one of the processes of cell division that can produce the new cells has allowed scientists to be able to artificially manipulate the natural process of cell division to produce what is called clones. So cloning is the production of genetically identical offspring from a single parent. So that's different than what you and I are, because we are genetically different from our parents, whereas cloning means it would be the exact same. So clones, as you can see down below, there are the cloned rabbits as well as the cloned horses on the right, meaning the sequence of their DNA is the exact same as as the organism that it was copied from. And this can be an advantage, but it can also be a huge disadvantage. So in reproductive cloning, the cloned embryos can be used to produce a new individual. Here you can see that if an animal is to be cloned, that embryo is going to be injected into a surrogate mother. In this case, it was a cow, and then she was going to have a Jersey cow because that was the cloned embryo. Scottish researchers used this method to first do a clone of Dolly the sheep in 1997. Dolly was the first adult mammal to be cloned, and in years after this, biologists have been able to clone many mammal species, such as dogs, cats, cows, and mice, as well as very rare and endangered species, so that can be helpful to have these clones available in the event that an animal would go extinct. In the use of cell cycles when it comes to therapeutic cloning, clone embryos can be used to produce stem cells. So stem cells are cells that have the capacity to turn into any cell that they want to, and that can be really advantageous when you're trying to treat certain diseases. So for example, in the bottom picture, we see an embryo, and the embryo is then going to have some stem cells removed. Those stem cells can be engineered to turn into blood cells, nerve cells, muscle cells, anything like that. So let's say that you are the patient in the far left, and you need new blood cells because you have a blood cancer. Well, we already have the capacity to produce your blood cells because we have your stem cells. Therefore, we can treat you when it comes to the cancer or the cancer type that you have. And so this is a really huge advantage, having this capability to treat and develop new cells from pre-existing ones by having exact clones. Where you will oftentimes see artificial cloning is in agriculture. It's very widely used. Many crop plants are propagated by simply placing a small sample, sometimes even individual cells, into a growth liquid. Those individual cells divide and develop into new plants, which then can be grown as normal. Many crops, such as things like orchids, oranges, and grapes, and other plants, they depend on that process. So you can see in the picture down at the bottom of the slide, they've taken a piece of this plant and they've then divided it and put it into an environment that has the growth liquid. And as a result, they can take that after it grows and plant it to become an adult. So sometimes you can see people doing this when they have a house plant, like an African violet. They can just clip off a leaf and it will then start growing on its own. So moving on to what we call gametes. Gametes are going to be something that's entirely different than cloning. because gametes are going to come from a sexual reproduction that results in a genetic variability. This is much different than cloning because what happens here is that the different sequence of DNA means that each one of us humans has a different sequence of DNA versus cloning, where cloning is the exact same sequence. So mitosis is the cell division that creates genetically identical sequences of DNA. And so we need another form of cell division that we're going to talk about shortly called meiosis. So what are gametes? So in order to prevent the doubling of chromosome number, sexually reproducing organisms have to make cells with a single set of chromosomes. This means that the two components that come together for sexual reproduction, they have a single set of DNA coming from each parent. So this means that sperm have 23, and this means that eggs have 23 chromosomes. So when you add 23 plus 23, that's where you get the 46 chromosomes. Therefore, gametes are also called sex cells. They're also haploid, meaning that they contain only half of the chromosome number. They essentially contain one copy. Then when the egg and sperm come together, they form a cell that is called a diploid cell. So here is that picture again of what's happening. In each of the adults, if we looked at all of their cells, they would all have 46 chromosomes, except for the male sperm is going to have 23 chromosomes, and the female egg is going to have 23 chromosomes. When those come together during fertilization, the egg and the sperm will come together to form 46 chromosomes, leading to a zygote. And a zygote has 2n, meaning 2 times 23, because for us as humans, n equals 23, which means that a zygote has 26 chromosomes in each of its cells. This then will grow into a human being, and the human being will develop into an adult, and should that adult choose to procreate, the process occurs over again. So every single cell in the human will have 46 chromosomes, except for in the sex cells, so either the sperm or the egg, and those will have 23 chromosomes. So the 46 chromosomes in your body are organized into 23 homologous pairs. The word homologous means that they are the same or that they look alike. So you have inherited 1/2 from mom and 1/2 from dad. Remember that for humans, n is going to be equal to 23. This means that in each of your gamete cells, you have half of the DNA, which makes them haploid. In the rest of your chromosomes, you'll have 46, or in the rest of your cells, you'll have 46 chromosomes, which means diploid. So you are a diploid organism because you have 2 times the DNA, 1/2 from mom, 1/2 from dad. Out of your chromosomes, 22 pairs are considered to be autosomes. The prefix auto means self, so this means a total of 44 of those 46 chromosomes are autosomes, or chromosomes that are going to be controlling yourself. Whereas the final pair are sex chromosomes, and these are going to be the ones that determine the sex. So females are considered to be XX, males are considered to be XY. On the right over here, you can see what chromosomes look like in real life. So in this case, because there's an X and a Y chromosome, we would be looking at a male. When we look at those 46 chromosomes in humans, one of the things we can do is take a picture. So this picture is called a karyotype, and a karyotype allows us to see the homologous chromosomes all lined up. And realistically, what you see is in the bottom right. You can see how they organize them, is they just take the biggest chromosome to the smallest. That's really how they do it. There's no order other than just to have them start with the biggest and end with the smallest. You can say, let's say hypothetically, it's not actually the case, but if we just said hypothetically, you can account for the fact that all of the left ones come from dad and all of the right ones come from mom. So therefore, if you counted all of the left ones, you should count 23. And if you count all the right ones, you should count 23. Because remember, you get half from mom, half from dad, and then 23 plus 23 would equal 46, which is each of these individual chromosomes. So here, just looking at this karyotype, can you tell if this one is male or female? So if we're looking at the sex chromosomes, we can see here that there are 2 X's, so this would be a female. And remember, you have these 2 chromosomes that are going to be your sex chromosomes. And then you have 22 pairs or 44 individual chromosomes that are your autosomes, which basically means your chromosomes just for yourself. Let's talk about another form of cell division, which is called meiosis. In meiosis, what's happening is that the cell division is going to be producing gametes, and these gametes then are going to be genetically different than their parents. This is a huge advantage because this means, for example, let's say that there was a disease that wiped out an entire population because it had the exact same sequence of DNA. Well, because organisms that have different sequences of DNA, they're more likely to survive because not every person or organism is going to have the exact same DNA. So remember, cells are going to be produced and form a haploid, so 23 chromosomes in eggs, 23 chromosomes in sperm. And just like mitosis, there's going to be phases that are part of this as well. So in meiosis, there are 2 rounds of cell division. The cell division names are the exact same as mitosis, but it goes through it twice. Just like mitosis, meiosis starts when the chromosomes are going to duplicate, and as a result, you get the exact scenario in which you have 4 haploid offspring cells, or remember the 4 cells that have 23 chromosomes to them, because they have half of the chromosomal number. So on the left side, it begins with 23, it duplicates to form doubles, and then it goes through 1 round of meiosis, the second round of meiosis, and then each one of these offspring cells are going to have 23 chromosomes. And you'll notice how all of them are different, and that is exactly what should happen. So these are the phases of meiosis, and you can see that they're the exact same phases as mitosis. It's just that we end them to indicate what number they're in. So that will often be indicated just by the letter I or 2. Sometimes there's superscript or subscript letters. So you can see interphase through telophase 1, and then prophase through telophase 2. And at the end of telophase 2, so at the end of meiosis, you should have 4 cells, which is what you can see over on the right, and each of them are going to have some genetic variation. So that's what we can see here. Mitosis and meiosis play a role in sexually producing organisms. So meiosis is what produces the genetic variability between the egg and the sperm. That's going to then create a zygote, and then that will turn to mitosis, which is where the cell is going to go through cell division to produce a human being, and then that human being will develop into an adult, and then that adult is going to be producing meiosis in the form of either egg or sperm. So let's compare mitosis and meiosis to make sure that we understand what's happening. So where does it occur? Meiosis is only going to occur in ovaries and testes, whereas mitosis occurs in all the rest of the body cells. When do you need meiosis? You only need meiosis once you are going through the process of reproduction, which begins at puberty. So for women, they finish up their meiosis pretty much when they hit menopause, which is when they're done ovulating. Men will continue meiosis for the rest of their life because they will never not produce sperm. Mitosis is going to last a lifetime. So you will continue mitosis all day, every day, throughout your life because you need to produce skin cells, hair cells, everything like that. How many cells come from each of these cell cycles? So in meiosis, there's going to be 4 cells, and in mitosis, there's going to be 2. In each of these cells in meiosis, they will be haploid, meaning that they will have 23 chromosomes. And in mitosis, they're going to be diploid. So that means that they will have 46 chromosomes. And then how many rounds of cell division occur? In meiosis, that will be 2, and in mitosis, that will be 1. A huge advantage of sexual reproduction is the fact that it leads to variation. And remember that this is different than cloning because cloning is the exact copy of the parent. So for organisms that do the cloning or asexual reproduction, they can do it quick, they don't need another partner, so that's a huge advantage for them. But the genetic variability is a big piece that they're missing. So again, if a disease came along and it infected one of the organisms that had a certain DNA sequence and it killed all of them, you'd assume that every organism that has that same DNA sequence would also possibly be killed. Whereas sexual reproduction allows for variation, ensuring that the survival of the fittest is who is going to survive. There are three main processes that are going to lead to sexual reproductive variation in organisms. The first one is called independent assortment. Then we have random fertilization, and then crossing over. So we're going to take a look at each of these. Independent assortment is one of the things that will lead to variation. So what happens is that the chromosomes will line up very randomly down the middle of the cell when it's going through the cell cycle. As a result, this is going to create a huge genetic variation. Independent assortment alone can account for 8 million possibilities of arrangements of chromosomes. Obviously, people do not have 8 million children, so therefore it's a really, really effective and creative way of generating genetic variability. So the idea here is if we happen to have, for an example, a specific sequence, so let's say it lined up, let's see, 1, 2, 3, 4. Let's say that it would be lined up that way. Well, in independent assortment, that means that it could line up with, say, 2, 4, 1, and 3. Hopefully that kind of comes across on your screen. So what this means is that it's going to lead to variation in the sequence of these chromosomes. So they're going to match up in a variety of ways. Random fertilization means that the egg and the sperm is going to be fertilized by the most optimal sperm. The probability that any one sperm will fertilize any particular egg is actually quite small. So in each of these sperm, there's 8 million possibility of chromosomes arrangements, and in the egg, there's 8 million possibilities of that chromosomal arrangement. So therefore, 8 million times 8 million means that there are 64 trillion possible arrangements of chromosomes. Again, huge, huge genetic variation because people are not having 64 trillion offspring. The third piece that leads to genetic variation is called crossing over. During crossing over, which happens during meiosis, the DNA is literally just swapping parts of it. And so those chromosomes create new sequences that you can see here in this picture or this drawing. We have the maternal chromosome in red and the paternal chromosome in blue, and they literally just swap. And so as a result, if you were to draw a dividing line between the sister chromatids in the chromosomes on the right, you would see that each of those 4 individuals are going to be different, and that leads to hybrid chromosomes, that leads to genetic variation. So even though those 3 items are very effective at creating genetic variation, they can also lead to mishaps. And when mishaps happen in the genetic sequence, we call this non-disjunction. And non-disjunction is when your eggs or your gametes or the chromosomes fail to separate properly. As a result, you may end up with more or less than that number of 46 chromosomes. Typically, the body does a very good job of vetting and making sure that if it's not the magic 46 or the magic number 46, then the individual would not be born. But there are some scenarios that non-disjunction has resulted in conditions in which the offspring can survive. So typically, zygotes that have an abnormal number do not survive, but occasionally they will. And sometimes we will see 1 extra or 1 fewer chromosomes as a result of that. So again, what should happen in the process of development, or a typical normal process, we have an egg that has 23 chromosomes, sperm that has 23 chromosomes. They come together, they form 2N, which means that they're going to have 2 times the magic number of N, which leads to 46 chromosomes. In non-disjunction, so that is when errors occur. For example, In the ovary cell here in the egg, it might have one extra chromosome, so 24, or one fewer, which means that it's going to have 22. And then that pairs together with the sperm, then that's going to generate a zygote. And the zygote then, in this case, is either going to have an extra chromosome or one less. So sometimes this will result in a spontaneous miscarriage. Other times the child will fully develop. So here is a condition in which having one extra chromosome is going to allow the zygote to most often survive. This is called trisomy 21. You also may know this as Down syndrome. All it is in these individuals is that they present with a third chromosome, and that third chromosome is a copy of the 21st chromosome, which results in Down syndrome. So all people with Down syndrome have this third copy of the 21st chromosome. They have often very similar characteristics, so short stature, heart defects, varying degrees of delays, sometimes susceptibilities to certain diseases. But regardless, they all have the same scenario going on with their chromosomes. It's just that one extra copy of their third chromosome. So non-disjunction can also affect the sex chromosomes. The previous one that we looked at was in the autosomes. So in sex chromosomes, that's going to refer to what can happen if you have a different variation of your X's and your Y's. Each combination of extra or missing sex chromosomes produces its own outcome. So if an embryo lacks an X chromosome, it cannot survive, but other abnormalities, such as having a single X or no Y or extra X's and Y's, have less dramatic effect. So down here you can see there are some examples where somebody has 3 X's. This is called triple X syndrome. This would be a female because all of them are X's. There's XYY, which is called Jacob syndrome. This is only going to be found in males. There's XXY, which is Klinefelter syndrome, again, only found in males. And then XO, which means that only one X chromosome appears. This is called Turner syndrome, and this means that those individuals are going to be sterile. So what you're seeing here is that these sex chromosomes, what's interesting is that the first 3, so the triple X, XYY, and XXY, sometimes these individuals don't even know that they have this scenario going on in their chromosomes until they go to procreate or to explain some underlying health condition that they have.